Hello everyone, welcome to this wonderful session. In this session we have to discuss Part 6, Chapter 2, Biological Classification. This part will include that is Ascomycetes, Basidiomycetes and Deuteromycetes. That is 3 classes of fungus we have to discuss here. Let's start with this Part 6 of Chapter 2. Now, Ascomycetes. Ascomycetes is known as sac fungi because the spores produced in this are in a sac-like, in a bag-like structure known as ascus, and therefore the name is given Ascomycetes. Ascomycetes, these are saprophytic, decomposers, parasitic or coprophilous. Coprophilous means that these are found on dung, especially on dung, which is a decaying organic matter. Ascomycetes, mostly these are that is multicellular, that is these are made up of hyphae like this, which are thread-like structures, and these thread-like structures, they have this septa they have. For example, Penicillium and Aspergillus and so many, most of the ascofungi, these are multicellular and made up of hyphae. Group of hyphae is known as mycelium. But a few are here which are unicellular, non-mycelial one. For example, yeast. You know, the yeast being unicellular and eukaryotic, nowadays in 5 kingdom classification, it has been kept there in kingdom that is Protista. You know it now. But as most of the characters of yeast, they resemble with other fungi, that's why we have to keep this yeast even in fungi, kingdom fungi. You see this mycelium, or it's better to call it hyphae here, that the mycelium, these are very well branched and these are septate like this. These are branched, septate and monokaryotic. Very important term this is. Monokaryotic means that each cell is having one that is nucleus. This nucleus may be of plus strain in one hyphae like this, or in other hyphae that nucleus will be of that is minus strain. So such mycelium, such hyphae is known as monokaryotic. Mono means one, karyon means nucleus. Each cell that carries one nucleus, and this is called that is primary mycelium. You have to remember this is primary mycelium, right? You have to see in time to come that is secondary mycelium will also be there. We will discuss in time to come. But here I'll give the reference of this. This is secondary mycelium. Secondary mycelium means each cell is having 2 nuclei. One is of plus strain and the other will be of minus strain. When the cell has 2 nuclei, that will be known as that is dikaryon, or that is known as that is secondary mycelium. Is it right? Secondary mycelium. Monokaryotic means each cell is having 1 nucleus. Dikaryotic means that is each cell is having 2 nucleus that is of different strain, plus and that is minus. Asexual reproduction here, that is important, that is carried out by conidia. Conidia, these are thin-walled and non-motile structure. You see that this is the mycelium here, primary mycelium. That is, a branch will come out, that will become aerial. Its tips will become globose, or that is rounded. And here, that is nuclei that will divide. Nucleus will divide. And here, that these nuclei will be transferred outer side. This structure is known as that is conidium. And in conidium, the spores are produced, asexual spores are produced, which are known as conidia. You see, conidia are, you are seeing here, these are thin-walled and non-motile one. Each is having 1 nucleus. And these conidia are produced outside the conidium. Therefore, these are exogenous. Exogenous means are produced outside, right? And this, one more specific point with this conidia, that is, these are arranged in a specific way. The youngest and the smallest one is close to conidium, and the oldest and the largest one, it is away from conidium. So this type of arrangement is known as basipetal arrangement. This is basipetal arrangement of, that is conidia, basipetally arranged. I may summarize. Asexual reproduction is by conidia. These are thin-walled and that is non-motile one. These are produced exogenously and these are, that is basipetally arranged. Monokaryote, that is, come to this now. Sexual reproduction, that is by conjugation, as in case of yeast, the two cells, two yeast with different strain, they will come close, they will make a conjugation tube and their cytoplasm will get mixed. It may be by gametangial contact, the detail is not required here, that as in case of penicillium. What happens? Gametangial contact in also, that is the different strain hyphae, they will make the gametangia and they will come in contact so that different nuclei, that is of different strain, two nuclei, they will become close and they will be brought closer to each other. Monokaryotic hyphae, as I told you here, this is monokaryotic hyphae. This is long-lived. That will remain, let the life cycle is for 1 year. For 11 months approximate, for a long time, that will be monokaryotic one, and dikaryotic hyphae, that is short-lived. Dominant one is primary mycelium, monokaryotic one, while the secondary mycelium, that is short-lived. Dikaryotic phase is short-lived as compared to monokaryotic one. The dikaryotic phase we can represent like this. That this is how that will be formed. You see that this is, that is one hyphae with plus strain, that this is other hyphae that with minus strain. That the two will come in contact and what will be now? That a cell will be formed which is having two nuclei, one of plus, one of minus strain. So this is known as plasmogamy between the two somatic hyphae, and result of this plasmogamy that will make a cell known as, that is dikaryon. So this dikaryotic phase we will represent it by, that will be presented N and N. This dikaryotic phase will represent as N plus N, and when the nuclei will fuse, that will be represented by 2N. So monokaryotic one, this is haploid. Dikaryotic phase, this is also haploid, and that is 2N phase, that will be diploid one. What will happen after that? That is after plasmogamy, that is karyogamy will not take place at once. This is delayed one, and karyogamy, that will be followed by meiosis. After meiosis, the ascospores will be formed, and these ascospores, as I told you in the beginning, they will be formed inside the bag-like structure known as ascus. Therefore, that is, the group is known as ascomycetes. In some of the cases, these asci which are formed here, that this is the dikaryotic phase. Dikaryotic phase, here the nucleus will fuse, that is zygote will be formed, and now this zygote will divide. What will be, that is ascospores will be formed, which may be 4, maybe 8 in number. This bag-like structure is known as, that is ascus. Now in some cases that the ascus are found in group, so many asci will be there in group, and that structure is known as, that is ascocarp. Ascocarp is formed in sexual life cycle which contains, that is asci. Now this group is very important. Its economic importance, we see some of the important examples. One is, that is Saccharomyces, that is known as budding yeast. Saccharomyces cerevisiae, you are familiar with that. Aspergillus flavus is known as guinea pig, that is of the plant kingdom, or known as, that is laboratory wheat. That is, you may be working with any fungus, with any bacteria in, that is microbiology lab. This fungus Aspergillus used to come just like a wheat. One important, that is genus we have, that is 𝕆𝕋𝔼𝔼. That is, this fungus has been used extensively in biochemical and genetic work. As Drosophila is being used in genetic work, in the same way 𝕆𝕋𝔼𝔼 is used frequently in Neurospora, is used in frequently in case of, that is, this is plant material, fungus this is. That's why it is called as Drosophila of plant kingdom. The important question, Drosophila plant kingdom, that is Neurospora crassa. Claviceps purpurea, you see, that is a fungus which causes a parasitic fungus. It causes agate of, that is rye, that is the source of LSD. Penicillium, different species we have. Penicillium notatum, that was reported first, from which that is antibiotics penicillium that was being obtained, penicillium. And nowadays Penicillium chrysogenum, from this fungus we used to obtain antibiotic penicillin at commercial level. Not only this, some of the edible ascocarp we have, that is Morshella. In Hindi we used to call it gucci, that is rich source of protein. Morshella. Morels, general, we used to call it as morels. This is having edible ascocarp, a good source of protein. And truffles also, layman language known as, that is edible tuber. These are also, that is a source of human food. You can see, dear children, here, this is the yeast we have here. Yeast, this is unicellular non-mycelial fungus. You see the budding is there. This is penicillium. This is these are the conidia, and this is conidium, and on conidium these are that is conidiospores, which are arranged, that is basically like this. This is the culture of penicillium, and this is well known. You must have seen in market also. This is Morshella, that is edible fungus. This is edible Ascomycetes. This is right. Come to the next group now. That group is, that is Basidiomycetes. Basidiomycetes is known as club fungi. Ascomycetes was ascus fungi. It's known as club fungi. And this is the fungus which is, that is commonly seen, especially in rainy season. You might have seen this type of fungus. This is that is edible mushroom. This is available in the market. You must have seen bracket fungi, this type of fungi. That is very common, that is rotten wood logs. This is puff balls, and this is the rust of wheat. On wheat leaf you will see that is rusty appearance, that is spots are there. That is four important, that is Basidiomycetes we have. That is mushrooms, bracket fungi, puff balls, and that is rust of wheat. Same way, they include, what they include? Basidiomycetes include bracket fungi, mushrooms, puff balls, rust and smuts of fungi, most commonly seen, and that is most advanced group of fungi. Is it? Once the question was there, which one is odd? Bracket fungi, mushrooms, puff balls, and morels. Morels, this doesn't belong to Basidiomycetes, that belongs to Ascomycetes. That's why that will become odd one. Which is a highly advanced group of fungi, that is Basidiomycetes. They produce club-shaped basidia, and on this, that is basidiospores are formed during sexual life cycle. That is tell you here, that is this is the monokaryotic hyphae. In the same way, this is monokaryotic hyphae with plus strain. This is the monokaryotic hyphae with minus strain. Monokaryotic hyphae, that is short-lived. In case of Ascomycetes, that was long-lived. Here monokaryotic hyphae, this one is short-lived. That is at the earliest, that is they will get fused, and this in the same way that the two somatic hyphae of different strain will come close, and they will make a dikaryon phase will be formed with plus and minus, that is nuclei. Dikaryotic phase, that is dominating one, that is long-lasting one. The difference you can see here in Ascomycetes. Come on, in Ascomycetes, if we compare here, let this is Ascomycetes, let this one is Basidiomycetes. Important question here is, that is primary mycelium, is it? And let this one is, that is secondary mycelium. In case of Ascomycetes, primary mycelium is, that is long-lived. Basidiomycetes, primary mycelium is short-lived. Is it? Here, Ascomycetes, primary mycelium is long-lived. In Basidiomycetes is short-lived. While secondary mycelium in Ascomycetes is short-lived. In case of Basidiomycetes, that is for a long time. This is dominating one. What happens after this? That is these two cells, this is plasmogamy. After plasmogamy, there will be what? There will be, that is karyogamy. After karyogamy, that the nucleus have been fused, that is zygote has been formed. Now there will be, you can see it here, just see, that is this structure have been formed. This is known as basidium. In this basidium, that is the zygote is there. Zygote will divide by meiosis. There will be reduction division, as a result of which, that is 4 nuclei will be formed. 1, 2, 3 and 4. 2 are of plus strain, 2 are of minus strain. In later on, that this will be shifted outer side, which will make the basidiospores plus, and these are the 2 will be of minus. Therefore, the basidiospores are formed outside, exogenously these are, and this structure on which these are born, this is known as, that is basidium. We see the same thing, basidia are club-shaped structure, and that is basidiospores are formed outside, exogenous these are, and these are formed in, that is sexual life cycle. That is, if the question is there, which is the best decomposer, that is Basidiomycetes fungus. These are the best decomposer of wood. Even the lignin, which cannot be decomposed by other microorganism, Basidiomycetes can decompose lignin also, as well as cellulose also. The important thing here, that is a few member, most of these are, that is saprophytic one. This is saprophyte, this is saprophyte, this is saprophyte. But you see, a few fungi are here from this group, which are parasite, and they cause well-known disease of plants, that is rust and smut. You can see this is the rust of wheat, right? Come to the next now, that in the same way, as I told you, secondary mycelium, dikaryotic mycelium, this is long-lived, I told you. Primary mycelium is monokaryotic, and that is short-lived. It develops by germination of the basidiospore. I can repeat it. This is basidiospore plus strain, basidiospore minus strain. They will germinate, mycelium will be formed. This is known as, that is primary mycelium, monokaryotic one, with plus strain, with minus strain. Then there will be what? There will be plasmogamy now. Is it? You know, that is one step in sexual reproduction. Plasmogamy will result in formation of, that is this dikaryotic cell. And now this dikaryotic cell, that will develop into this dikaryotic mycelium, which is having 2 nuclei. One is of plus strain, one is of minus strain. That is, once a cell is formed, dikaryotic cell, other nearby cell will also become dikaryotic one, and a phenomena is there known as clamp connection. Clamp connection, by this, that is, that nucleus of opposite strain will be distributed to the nearby cell, right? Vegetative reproduction, that is by fragmentation. Simple thing. This is hyphae that will divide into pieces. Each will be able to develop into the new one. Asexual, this is important one. Asexual spores usually are absent. Asexual reproduction is not reported in Basidiomycetes. Sex organs are absent. Plasmogamy is brought by fusion of 2 vegetative or somatic cells of different genotype. The same thing. This is called somatogamy. Karyogamy takes place in basidium, right? And basidiospores are produced outside of the basidium, exogenous, and the fruiting body of this is known as basidiocarp. You see, children, that this is basidiocarp. This is basidiocarp. If you enlarge the structure of this basidiocarp, it will be like this, that here these are the, that is, this is the group of mycelia. On both sides of this, these are the basidia here, and on this, that is basidiospores are born. You must have seen this umbrella-shaped structure. This is basidium of agaricus, that is edible mushroom, right? Now, one more important point here we have. This is very, very important one. In Basidiomycetes, if we compare, like this is Phycomycetes and this is that is Basidiomycetes, the sexual reproduction as one goes from lower fungi towards higher fungi, that is complexity of sexual reproduction, complexity that decreases. Very important point. That is sexual reproduction Advanced sexual reproduction is found in 𝕍𝕍𝔻𝔼, and that is very simple type of sexual reproduction by somatogamy is found in 𝕆𝕍𝕻𝔻. 𝕆𝕍𝕻𝔻, it doesn't have sex organs. Some important fungi of the group. One is that is Agaricus campestris, edible mushroom. Easily you have seen in the market, a good source of protein. Astilagotritis, this is known as loge smut of wheat, a parasitic fungus this is. Similarly, Puccinia graminis tritici, black rust disease of wheat. You are seeing that this is the rust of wheat. And one important fungus we have, that is Amanita cicerea. It's known as toad stool. This is a poisonous fungus. Come to the next now. That is the last class of fungi we have, Deuteromycetes, or known as that is fungi imperfecti. Why fungi imperfecti? Because in all the groups you have seen, there was sexual reproduction. No doubt, sexual reproduction. That is complexity, as I told you right now. Complexity, that is complexity that keeps on decreasing. That is complexity that is in 𝕍𝕍𝔻𝔼, that is highly complex type of sexual reproduction, while in case of 𝕆𝕍𝕻𝔻, the sexual reproduction was simple one. Interesting point here is, in Deuteromycetes, that is sexual reproduction is not reported yet. Sexual reproduction is not reported or is absent. Therefore, these are not perfect. It's known as fungi imperfecti. The question here is not reported, is not found, or is absent. It's better to call not reported. As and when sexual reproduction will be reported, that is the fungus which are kept in a Deuteromycetes, as and when that is sexual reproduction is discovered, the Deuteromycetes either will be shifted to Ascomycetes or will be shifted to that is Basidiomycetes, depending on the type of mycelium. You can read it out now. That is these are saprophytic fungus, and they help in mineral cycling by decomposing litter. A few are parasite also. Mostly these are saprophyte. Important question. Asexual reproduction, as usual, this is by conidia, as you have seen in case of Ascomycetes. That is, conidia will be produced outside this conidium and are arranged basipetally. That is sexual reproduction. Very important point here. This is a very important point. That is, sexual reproduction is unknown, not reported yet. Once the perfect stage or sexual reproduction is discovered, that will be shifted to Ascomycetes or Basidiomycetes, depending on the type of mycelial structure. Is it? You know, mycelium or hyphae of the Ascomycetes and Basidiomycetes, they differ. They have, no doubt, both of these, they are having this septata, septum they have in between the cells, septum is there, but type of septum differs on the basis of this septum structure. That is, this Deuteromycetes will be shifted either to Ascomycetes or to Basidiomycetes. This group of fungi, mostly they are just saprophyte, as I told you, but a few fungus are, that is, parasitic also. Alternaria solanae, early blight of potato. Remember this question. There is one that is late blight of potato. Late blight of potato. Can you remember? Late blight of potato, that is caused by Phytophthora. Can you remember it? That belongs to, that is, Oomycetes of, that is, Phycomycetes. This question, this is Alternaria solanae, this is early blight, that was late blight of potato. Colito trichum falcatum, this is red rot of sugarcane. You must have seen sugarcane sometime, that is, yellow strips are there. If you that split this, that is internode of this. Then Helminthosporium oryzae, brown leaf spot of rice, Bengal famine in 1942-43. It was caused because of this fungus, Helminthosporium oryzae. And Trichoderma, a very important fungus, that is from Trichoderma, that is bioactive substances is obtained, bioactive substances is obtained. This is about Deuteromycetes. In that way, we have finished, that is fungi, its general characteristics. Then we have seen the groups of fungi, Phycomycetes, ascomycetes, basidiomycetes, and in the last year I have seen 𝕍𝕀𝔻𝔼. It's okay. This is for this lesson. Remaining part of the chapter we have to do in next part of this, that is part 7, and after part 7 we have to discuss questions, NCERT exemplar, and questions which had appeared in NEET examination. Thank you. Have a nice day.